Hi. Okay. I'm back and I'm going to teach you all about heredity. Since I already went over the review sheets and all the answers to them, I think I'm just going to go through the chapter and then go through all the PowerPoint slides and then explain them so that you can hear this as another way of studying. I try to do it with the PowerPoint instead of seeing my face, seeing the PowerPoint, but that's how it didn't record last time. So I'm just going to talk to you. So here we go. The whole entire chapter starts with Gregor Mendel, and Gregor Mendel was the guy who studied the pea plants. He was a monk, and as he raised the pea plants, every single time he saw them, those tall plants were always making tall plants, and short plants were always making short plants. He noticed that there was always these two different traits, either tall or short, green or yellow, round or wrinkled, all these different traits. There was two different ones, and they always were getting passed down. So he wanted to know, well, what happens if I mix them? So if I mix a tall plant and a short plant, what will happen? If I mix a yellow plant and a green plant, what would happen? And normally we would think, okay, well, a tall and a short plant, they'll probably give us a medium plant. But that's not what happened. Instead, the tall plants and the short plants all made tall plants. And from that idea, he made up this principle of dominance idea. First, he said that all these things that are getting passed down must be things, so he called them genes. So a gene is an actual thing that is passed down in your DNA to your offspring. The different versions of those genes are the alleles. So height, for example, is a gene. The allele could be tall or the allele could be short. Color of the pea is a gene. It could either be yellow or it could be green. Those are two different alleles. So gene is the thing that's passed down. Alleles are the different versions of those genes. And he noticed that always there was only one that came out. Out of yellow and green, they all were yellow. Out of round and wrinkled, they were all round. And so the idea of dominance is that these different alleles, one allele is dominant over the other. It wins. It shows up, and that's the one that's going to be expressed. Instead of a combination between the two, you only get one. So that was his principle of dominance. That was his first idea. So then he went on and he wanted to figure out, well, I had a tall plant, I had a short plant, and they all came out tall. So did that tall gene just go away? Did that allele just go away? What happened to it? So he took those generations of tall plants, they were all tall, and he crossed them together. And what happened was they both had an allele from the short plant, so they both carried that short allele, so they both passed it down, and about a fourth of all of their offspring were short. So this is a super important thing because this shows segregation. This means that, yes, you have two alleles. Every single organism has two alleles for every gene because you get one from mom and one from dad, but you only pass down one. They segregate from each other. So during meiosis, we had homologous pairs that were separating from each other. That's that separation. Half the sex cells end up getting the one allele, half the sex cells get the other allele. That's segregation. That's a good, important thing to know. That's why two tall plants could make short offspring because they have those alleles and segregation causes them to be passed down by themselves. In addition to that, it's also dominance and recessiveness because the only way the recessive trait came out was because there was no dominant allele. So know that as a question. How come that is possible? Segregation and the principle of dominance. Okay. So he did this thing. He figured out what you can do with all these plants is basically use probability. And so that's where Punnett squares come in. Punnett squares, what they're showing you is the probability of an offspring to look a certain way based off of their parents' genotypes. So important things you need to know. Genotype versus phenotype. Do not get this wrong. If I ask you for genotype, G is letters. I want letters. I want capital T, lowercase t, lowercase t, lowercase t. If it's asking for phenotype, it's asking what they actually look like. Phenotype, physical characteristic. What do they look like? Are they tall or are they short? So if I ask you a question and ask me for phenotypes and you give me big T, little t, it's going to be wrong. So make sure that you don't mess that up. So Punnett squares, hopefully you all can do a Punnett square. If I tell you a heterozygous plant, it means they have a big T, a little t. If I tell you a homozygous dominant, two big T's. If they are homozygous recessive, they are two little t's. I could also tell you if the tall is dominant and short is recessive, I can just tell you cross a short plant. You should know that a short plant, the only way to be a short plant is to be little t, so you know the genotype right away. So there's a couple ways I can say certain things, and you should be able to figure that out based off of what I say. So if I asked you to do a Punnett square, you should be able to give me the genotype ratio and the phenotype ratio. Genotype, you do the two capital letters, so how many are big T, big T? Then how many are heterozygous? Big T, little t. And then how many are recessive? Little t, little t. Phenotypic ratio, there are only two for the phenotypic ratio. In this case, big T, and big T, little t both give you tall plants. So both of them are the same phenotype. So how many are tall and how many are short? Okay, so those are all the little simple Punnett squares, only 4 boxes, nice and easy. You should be able to do those. I look like I have a black eye, don't I? I don't know why. Okay, so another thing he figured out, he said every time he saw a yellow and round pea, it always produced yellow and round peas. So he wanted to know, well, are these traits linked together? Do they get passed down together? If you have brown hair and brown eyes, are you passing them down together? So what he did was he made that big, huge cross. He looked at 2 genes at the same exact time. So now we're looking at the color and we are looking at whether it was round or wrinkled. And when he crossed them, so they were all round, they were all yellow, they were all green, they were all wrinkled. He first time crossed them, they all came out yellow and round because those were the dominant traits. So it makes sense. They all were yellow and round. So then he said, okay, well, if I cross these again, what will happen? That's where that big, huge cross comes in. Big, huge cross where you take big R, big Y, little y, and you find out all the combinations. If I give you big R, big Y, little y, like that, you should be able to tell me that there are 4 different combinations. I can't even do this backwards, but first, outer, inner, last. 4 different possible ways. What that's telling me is that there are 4 possible sex cells that can come from this organism. And you cross them with the other organism that looks exactly like this. And the ratio is always 9:3:3:1. 9 of them are all yellow and round. 3 of them are round but green. 3 of them are yellow but wrinkled. And then 1 of them were round or wrinkled and green. So you don't even have to do that. If the question asks you to cross 2 organisms that look like this and tell me what the ratio is, you know it, 9:3:3:1. That's only, though, if both of them are heterozygous. If I tell you now, here's another question. If I tell you that an organism looks like this, big R, big Y, and you cross them with no matter what you cross them with, you don't have to do a big square. Why do you not? Because the only thing that this guy can give his offspring are a big R and a big Y. So no matter what, every single offspring, it doesn't matter what the other 2 letters they have are from the other parent, they're going to have at least, there we go, 1 big R. So they're all going to be round. And they're going to have at least 1 big Y, and they're going to be yellow. Save yourself time. You don't have to do it. All round, all yellow. Okay? So that's why those big factor crosses are there, to prove to us that your genes aren't linked together. And that's independent assortment. So when the question comes up with a huge box and says, what is this showing you? It's showing you that your genes are not linked together and they independently assort. When meiosis happens and all of your chromosomes line up, they all completely line up random and things don't have to get passed down together. Independent assortment. Okay? Don't confuse segregation and independent assortment. Segregation is that one gene, you have those 2 alleles, they separate from each other. You only give one or the other. Independent assortment is the idea that all the traits you have are not linked together. They can be randomly shuffled and randomly passed down. Okay? So those are all basically his big principles. What we need to talk about now are all the exceptions to those principles. So everything that he said about that dominant stuff, sometimes there are variations. The first variation is incomplete dominance. Incomplete means nobody wins. If nobody wins, it's a tie. Tie will give you a blend. So if I tell you that red flowers and white flowers, you should know those are 4 o'clock flowers. They're called 4 o'clock flowers. If they're crossed, they will all be pink. You blend them together. Now, if the question says, oh, if the 4 o'clock flowers instead followed codominance, what would they be? Codominance, they both win, so they both show up. They both show. Think of that in your head. They both show. You see red and you see white, you see speckled. No matter what it is. If I tell you it's cows, or I tell you it's chickens. If it is incomplete dominance, you blend the 2. So black and white makes